Ericulture is defined as the scientific rearing of the eri silkworm.
It has a scientific name Samia reseni and is used for production of silk.
The speciality of ericulture is that the silkworm, Samia species, is raised on a plant called castor.
It is a type of non-mulberry silkworm, and this silk is also known as peace silk or ahimsa silk.
Because in this, we take the silk after the moth has emerged. We take the cocoon for the spinning after the moth has emerged. So the primary host plant for eri silkworm cultivation is castor, and the secondary host plants are caseru and tapioca. This here is one of the plants that we use for agriculture. This is castor, and here are the leaves. You can see these are the leaves of castor. Right now, all the leaves are on the top. All of these are on the top. So this plant is the primary food source.
Life cycle of Ericulture. Ericulture shows complete metamorphosis and consists of 4 stages. The egg stage, that can be in between 4 to 6 days. The female moth lays eggs in clusters. These eggs are kept under suitable conditions for incubation and hatch. Within 4 to 6 days, they become into tiny larvae. Larval stage, that can be in between 20 to 25 days. This is the most active feeding stage. The larvae pass through 5 instars.
Stages from 1st to 5th instar larva.
They mainly feed on castor leaves obtained from Ricinus communis. During this stage, regular feeding is essential.
First instar larva were fed 3 to 4 times, morning, afternoon, evening and night, and bed cleaning was done once or twice by dusting of lime by using muslin cloth. Second instar larva were fed 3 to 4 times, morning, afternoon, evening and night, and bed cleaning was done once or twice by dusting of lime by using muslin cloth. Third instar larva were fed 4 to 5 times, morning, bed cleaning, afternoon, evening and night. Bed cleaning was done once or twice depending on excreta by transfer of worms into another tray by dusting of lime by using muslin cloth. In fourth instar larva, we fed it 4 to 6 times, morning, afternoon, evening and night, and extra time during day as per the need. Bed cleaning was done once or twice depending on excreta by transfer of worms into another tray by dusting of lime by using muslin cloth. Identified different from fourth instar larva by creamy body color, and when they tend to start cocoon formation, they stop eating, try to get into a corner, and silk thread laden with sericin start coming out of their mouth. Fifth instar larva are fed 6 to 8 times morning, afternoon, evening and night, and extra time during day as per the need. Bed cleaning was done once or twice depending on excreta by transfer of worms into another tray by dusting of lime by using muslin cloth. Molting stage. It is a transition stage between two instars of larva, during which larva avoids to eat leaves, but the culture is still fed with few leaves just in case few silkworms out of many came out of molting earlier than the others. At the end of molting, silkworm casts away its older external layer of skin and grows in size.
Then pupal or cocoon stage, that can extend from 10 to 14 days. The fully grown larva stops feeding and starts spinning a cocoon. In airy silkworm, the cocoon is open-ended, which is special feature. Inside cocoon, the larva transforms into pupa. Then adult stage, that can be in between 4 to 5 days. After development, the adult moth emerges from the cocoon.
It is not fed and its main role is reproduction by pairing to lay eggs. Then this life cycle of airy silkworm is repeated.
Rearing of airy silkworm. Now coming to the rearing process.
During rearing of instars of larvae, a constant range of temperature and humidity is maintained inside the rearing chamber.
Important practices include feeding fresh castor leaves regularly, cleaning the rearing bed, proper care during molting, lime treatment for maintaining hygiene, then disinfection. To prevent diseases, disinfection is using 2% formaldehyde and potassium permanganate.
Mounting.
When larvae becomes mature, they are transferred to mountages for cocoon formation. Mountages can be of plastic, wooden, or made up of leaves. There are some precautions that should be taken during rearing and while entering the lab, including cleanliness, disinfection, good ventilation.
When talking about agriculture distribution in India, among non-mulberry silk producers, Assam, famous for Moga and Eri silk, holds a significant share of northeast production. Agriculture is primarily done in Assam, Bihar, Manipur, Meghalaya, Nagaland, and West Bengal. While South India is the leading hub, Karnataka, Andhra Pradesh, Tamil Nadu, for mulberry. Objectives of learning agriculture: master sustainable host plant management, learn non-violent ahimsa rearing techniques, developed skills in post cocoon value addition, create market access for ethical textiles, hence the business opportunity. Agriculture at ANDC was quite fun, including learning fun done during rearing practices, leaf collection, etc., and arranging lab facilities. Thank you.